we left off talking about structure and function, or talking about the structure of an amino acid. All right, an amino acid structure ends up being central carbon. What's on either end? Central carbon. What's on an amine group? NH2. What's on the other end? Right? C double bond O OH. Okay? Central hydrogen. And then what's this? The R group. The R group is being your difference between amino acids. All right? Now we moved on to we talked briefly about proteins and their difference in structure. All right? We are going to go through the different types of structure, functional proteins today. Structure and function. Here's an example of structure, different ways to look at it. We draw proteins in what we call a ribbon structure. You may see them like that, which shows you some secondary structure. Right? You see an alpha helix right there. These turns are alpha helix. This right here is what we call a beta sheet. Okay, parts of secondary structure. This is a space-filling model. It gives you an idea of globular structure, tertiary structure, how that's folded onto one another, okay, and what that protein may or may not do. All right. So, primary structure. First, structure is directly related to the sequence of amino acids, whether it's alanine, leucine, isoleucine, glycine, tryptophan, whatever it might be. Okay? That is your first level of structure. What amino acid is placed where in the sequence? Okay? Well, starts at methionine, then goes through amino acids 355. That sequence of 1 through 355 is your primary structure. The sequence of amino acids 1 through 355 is important for secondary and tertiary structure as well. All right? And its structure and function. All right? So here's showing a direct relationship between structure and function. Here you have an antibody protein on one side and the protein from the flu virus on another. The interaction is direct, right? The structure of the antibody, right, directly reflects the structure of the protein from the virus because that is what it is going to recognize, right? It will bind to this, form the antigen. How many times can I say antigen? Antigen antibody complex, which will elicit an immune response. Okay? But structure and function directly related. All right, so your 4 levels of protein structure. Okay? There's 3 superimposed level of protein structure called primary, secondary, and tertiary. All right? The 4th level arises when a protein consists of more than one polypeptide chain. So, first level of structure is the chain of amino acids. The second level of structure, secondary structure, is how those amino acids fold into a stable form chain. Tertiary is now that folded chain folds upon itself to form a globular protein, and that is where a single polypeptide chain ends. All right? Whether or not that is a functional protein depends upon the function of that protein. All right? If the protein does not require other subunits to interact with it, that tertiary structure is the final level of structure for that protein. Okay? In some cases, such as in hemoglobin, you need other subunits come together to form a functional protein. All right, we're going to go through each one of these levels so you don't have to worry about, writing them down feverishly. All right. Each protein has a unique sequence of amino acids, 1 through 355. In that order determines protein A. Protein B may be amino acids 1 through 200. Amino acids in a different order than the first 200 of protein A, that determines protein B. Okay, and so on. All right. Secondary structure, it's found in most proteins, consists of coils and folds in the polypeptide chain. We'll talk about alpha helices and beta sheets, right? And that interaction between parts of the amino acid chain. And your tertiary structure is determined by fitting in those R groups, right, that kind of stick off the side of the amino acid chain. We have to fit them into a stable form, forming a globular structure, right? So they could be twisted, turned, etc. All right. Those interactions between R groups actually can stabilize proteins as well, and helps determine their structure properties. And then quaternary structure involves a second polypeptide chain that will be utilized to form your functional protein. So here's primary structure, okay, amino acid end, the amino end, the carboxy end. All right, this is primary structure of transthyretin, a protein, and you have proteins, glycine, proline, threonine, glycine, threonine, glycine, glutamate, serine, lysine, cysteine, proline, blah, 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 all the way down to glucine at the end, or glutamic acid at the end. All right? It is just the sequence of amino acids. Pretty straightforward, right? Wake up. Everyone's awake. Here's the overall structure. Now, secondary structure involves hydrogen bonds forming between parts of that amino acid, forming a more stable molecule. Okay? Parts of that protein become stable in a certain form. Forms this helix. You have I'll enlarge this. You have hydrogen bonds forming between parts of the protein chain, between two separate amino acids in different parts of the protein chain. Okay? That's in one area of the overall protein. Okay? In the other area, you have other hydrogen bonds forming another type of configuration called a beta sheet. Okay? This is an alpha helix. This is a beta sheet. Okay? They are not exclusive from each other. In one protein, such as transthyretin here, you will have parts of the protein will be set in an alpha helix. Other parts will be set in a beta sheet. It is a stable, lowest or stable conformation for that part of the protein. Okay? It is not exclusive to each other. All right? So you have parts of the protein that will fold into these helices and sheets, and then the overall protein folds again, forming your globular structure, what you see here. Okay? Here you have interactions between the two, between amino acids. All right? This is part of your tertiary structure. What you have here are examples of interactions between two R groups.
It's for biolab.
They have sea monkeys. Okay. All right, so you have hydrogen bonds at first forming between two amino acid groups. That helps form a beta sheet. Then you have interactions between the R groups, whether it be hydrophobic interactions. You could also have disulfide bridges, an actual covalent bond that's formed between the two, or even ionic bonds that are formed between ionic interactions between two R groups, like depending upon the R groups that are present. So what would happen if this amino acid was changed inside the sequence? Let's change it from glutamate or whatever it is to valine, or to an amino acid that has a ring here. What's going to happen to the overall protein as a whole? The shape will change. Why? No longer are you making this ionic interaction. No longer are you having that R group interaction that changes the globular shape of the protein. If you change this amino acid, no longer have that sulfur interaction there. Okay? You can no longer form a disulfide bridge. That changes the shape of the protein, changes the strength and overall structural soundness of the protein. All right? You may be able to change, say, amino acid here and not have a big difference, right? Because it's not directly involved in the shape of the protein. All right? This is an example of, this is collagen. Collagen are helical proteins that function in subunits. All right? So you have a trimer. 3 subunits work together to form the functional collagen protein. This is an example of quaternary structure. Right, and this is actually showing your secondary and tertiary and quaternary structure in one picture. All right, quaternary structure involves, 1, 2, and 3. Your blue, red, and green. Right, trimers, your monomers as a trimer, your alpha helices as a secondary structure, and then the whole thing together, quaternary structure. All right, another example is your hemoglobin. Okay, you have subunits. Right, there are actually 4 subunits present there. Okay, 2 alpha subunits, 2 beta subunits. Each of which binds an oxygen through the heme group. So you have 4 heme groups. Right, 1 molecule of hemoglobin can bind 4 oxygen atoms. All right, so this is an example of quaternary structure. Questions on primary, secondary, tertiary, or quaternary structure of proteins. All of them are equally important for shape and function of the protein itself. All right, an example for sickle cell disease. All right, what is sickle cell disease? Anyone? Cells are shaped. The cells are deformed. Why are the cells deformed? What does it have to do with? It substitutes an amino acid. Okay? Sickle cell disease has to do with red blood cells. Okay? Hemoglobin. You substitute one amino acid within the amino acid sequence of hemoglobin, and it changes the entire function and shape, not only of this protein, but eventually of red blood cells. Okay? So, you end up changing a little bit of primary structure. Notice amino acid 6 is a glutamic acid. Right? We switch that to valine in those who are affected with sickle cell disease, and now that changes the structure. So the normal secondary and tertiary structure gives you a globular protein, allows for interaction. However, when you you replace the glutamic acid with the valine, you end up with exposed hydrophobic regions, a different shape of secondary and tertiary structure, basically altering the function of the hemoglobin protein itself. All right? Therefore, molecules crystallize into a fiber and end up giving you a different structure of the red blood cells themselves. All right? Prevents them from adequately binding oxygen. All right? Look at the sickle cell disorder. Those 2 subunits that are misshapen cannot adequately bind oxygen. All right? So you have a deficit of oxygen, of the potential binding for oxygen. Okay? What else determines protein structure? All right? Proteins, carbohydrates, lipids, nucleic acids are optimized to work at specific pH within your cells. Okay? Changing at a specific temperature within your cells. Changing them, right? Changing temperature, pH, will alter the interactions between amino acids and between R groups, hydrogen bonds are altered, which will effectively change shape. Okay? That leads to denaturation of proteins, which is effectively disrupting a protein's native shape. Okay? Now, cells, you get a fever. Your fever goes up to 104, which is right about the edge of where things are going to get really, really bad. All right? Your cells are not happy at that elevated temperature. What's going on in your cells? Proteins at that point are denaturing. Okay? So now they're changing shape. They're losing some of their function. I give you ibuprofen, Advil, Tylenol to bring the fever down. Your temperature comes back to normal. What happens to those proteins that have changed shape in your cells? What does your cell do with that? Anyone? Cell has two options. Option number one, get rid of all the misfolded proteins completely, break them all down, and build new ones that are folded the right way. Okay? Option number two is to somehow refold the misfolded proteins in the right way. Which one do you think is most efficient? Refolding, starting over. Refolding, both are correct because it depends upon the protein itself. All right? For the most part, a lot of proteins can be refolded, and your cells have special proteins that will bind to misfolded proteins and refold them. They're actually called heat shock proteins because they're induced by changes in heat. It's like a fever. Okay? But some proteins are so important that they can't be refolded, that you have to start anew. So both of those actually occur. The most efficient process, right, and most speedy is the refolding, all right, what we call renaturation. So cells will hopefully, normal protein denatures, whether it be because of pH or temperature or whatever reason, enzymatic disruption. A lot of times that denatured protein can be refolded or renatured back to the normal protein, depending upon the protein itself, which protein we're talking about, right? Because some of them are not refolded. Questions on denaturation? All right, so pH, anything that will disrupt the interactions between amino acids can effectively change the shape, ultimately change the shape of a protein. All right, so protein in the cell, you don't have to know what they do. This was part of your chapter, so I wanted to mention it. They use X-ray crystals, is using a protein that's isolated, exposing it to X-ray radiation, and then they can look at the structure of the protein as its native structure. Now we have molecular tools, bioinformatics, that will predict the structure of a protein, okay, and how it is. Within the cell, it is very much controlled. How a protein folds is controlled as protein synthesis is ongoing. We're going to sneeze, excuse me. Questions on proteins and structure. The 4 levels of structure and what they mean. Nope. Okay, good. The next part is, the next macromolecule is nucleic acids. All right? Nucleic acids, including DNA and RNA. All right? These are important for the storage and transmission of genetic information. All right? Hereditary information. Okay? What's the importance of genetic information? Well, the amino acid sequence that makes up proteins, which then carry out all the functions of the cell, are stored in the genetic information, in genes. Each gene, most of the time, codes for a protein. So one gene, one protein hypothesis. Okay? By saving that genetic information, you ensure that subsequent generations of cells can code and produce that protein to do that function. Okay? That's the importance of genes. Right? Genes are made up of DNA, right, which is a nucleic acid, made up of monomers called nucleotides. So let's review. What type of monomers make up carbohydrates? Monosaccharides. All right. Which type of monomers make up lipids? Fatty acids, glycerol, and what's the starting point for some other lipids? Cholesterol. Okay. Which type of monomers make up proteins? Amino acids. And then which type of monomers make up nucleic acids? Nucleotides. All right. We're building. I like it. All right. Two types of nucleic acids. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Right? Differences between the two. DNA is the genetic material. Right? Stores information. Okay? It is a double helix. And we'll go through the differences in the nucleotides in a little bit. All right? DNA provides for its own replication. Okay? And when we go through the structure of DNA in about a month, maybe a little sooner, when we go through the molecular biology, we'll go through how DNA is replicated and what parts of it is important for its replication. Okay? And then DNA also directs the formation of the synthesis of RNA, particularly messenger RNA, right? Messenger RNA relays the code for each gene to the cytoplasm, which then can lead to protein synthesis at the ribosome. All right, so here's a general schematic of DNA and RNA. DNA is found in the eukaryotic nucleus, not necessarily in the prokaryotic. There's no nucleus in prokaryote cells. All right, moves out of the nucleus to the cytoplasm, binds the ribosome. Proteins then are formed off of that messenger RNA. The components of nucleic acids, all right, are nucleotides. They have three parts. All right, there's a sugar, depending upon which type of nucleic acid we're talking about, which changes the type of sugar. Okay, it is a pentose sugar regardless, 5-carbon sugar. All right, so we have a pentose sugar, nitrogenous base, which would be A, C, T, G, U. You all know those, right? Adenine, cytosine, thymine, guanine, uracil. All right, and then also has one or more phosphate groups, depending upon which level of phosphate, which level of nucleotide we're talking about. Okay, and then what difference in terminology? The portion of a nucleotide without the phosphate is called a nucleoside. So, pentose sugar bound to your nitrogenous base is a nucleoside. All right, first DNA is double helix. Actually, we're going to show. I'm going to draw the pentose sugar. All right, here's a nucleotide. Here's your pentose sugar. Depending upon what happens at that carbon, whether it's an OH or just an H, determines whether it's a ribose or a deoxyribose. Okay, at this one carbon, you have your bases. G or U, then you have another carbon here, and then this is where your phosphate groups are. All right, so phosphate, sugar, that's a 2, and then 3, your base. That's your structure of your nucleotide, 3 parts. All right, which all 3 put together give your subunits of DNA, RNA, nucleotide subunits. questions on structures of nucleotides? So for the structure of the complex together, you have DNA on the left and RNA on the right. DNA is a double-stranded helix, two strands held together by hydrogen bonds between the base pairs, right? Adenine, thymine, cytosine, guanine. What don't you see in the RNA on the right? What is missing? Thymine and a second strand. Okay, so there's no RNA, there's no second strand of RNA, right? RNA is single-stranded, and thymine is removed and replaced with uracil, the other base, right? So uracil, that's the U, right? And thymine and DNA. So already differences between DNA and RNA. DNA is double-stranded, deoxyribose sugar, presence of thymine, right? N is the genetic material. RNA, single-stranded, uracil instead of thymine. It is the messenger, right? Codes for a gene. And ribose as a sugar. Here's another picture of your nucleotides. Right, so here's the nucleoside. Is this part. Right here is a nucleoside. Structure of your bases. T in DNA, uracil in RNA. Ribose, the difference between ribose. And you see the difference in the two of them are what happens at that second carbon. Okay. Determines whether or not you have a OH or an H is the difference between deoxyribose and ribose. Questions on structure of these nucleic acids and the differences between the two of them up to there. All right, so cytosine, thymine, uracil, adenine, guanine. We went over that. Thymine's only RNA. So this, I think the next couple slides are just writing through what we've already discussed. Kind of get ahead of myself there. All right. Important to note the structure of deoxyribose and ribose. Okay, and the distinction or the numbering of them. Okay, here's your pentose sugar. Okay, the one carbon is the one that's attached to the base. All right. Number 2 is the one that's going to be either H or OH, depending upon if it's deoxyribose or ribose. The 3 carbon, right, a 3 prime, in nucleotides has an OH. That's going to be the linkage between the subsequent nucleotide. Okay, this is 4, and then 5 is another up here connected to your, or bonded to the phosphate group. Okay, that's the basic structure of a nucleotide. This is your 5 carbon, 3 carbon. Okay, important parts. The 5 prime. The prime is just used to dedicate what we nomenclature, right, numbering. The 5 prime carbon is attached to the phosphate. The 3 prime carbon has an OH on it. Okay, and if the OH is there, if it's free and not bound to a nucleotide, it can incorporate another nucleotide. All right. In order to incorporate more nucleotides and add subunits, you have to have a free 3 prime OH. Because what's going to happen? How are these going to be added together? Through which type of reaction are you going to add nucleotides together? Dehydration synthesis. All right, so you're going to have that OH. Those two are going to work together. All right, HOH. Water's going to come out, and you're going to produce what's called a phosphodiester bond between that 3 prime carbon and the 5 prime phosphate of the next nucleotide. Okay? That's what we're talking about here. So your adjacent nucleotides are joined by covalent bonds that form between the OH group on the 3' carbon of one nucleotide and the phosphate on the 5' carbon of the next. This creates your backbone, what we call a sugar phosphate backbone. Because if you went down a rung of one of the strands of DNA or RNA, the backbone, phosphate, sugar, phosphate, sugar. So it looks like this. Here's a sugar, phosphate to another sugar, right? Phosphate, and this is a chicken scratch drawing, but you get the idea, right? And all of the nitrogenous bases will be orientated to one side, away from the sugar phosphate backbone. Okay? The sequence of bases along the appendages, or the opposite side, opposite the sugar phosphate backbone, important for the function of what they are. For DNA, that's the sequence of bases represents the code for the gene. For RNA, it's important for which amino acid gets stuck in the. All right, so the structures. All right, RNA, single polypeptide, polynucleotide chain, single-stranded. DNA's double-stranded helix, which we call as anti-parallel, meaning one side, one strand runs 5' to 3', top to bottom. So 5' carbon, right? 3' phosphate, and ends off with the 3' OH. God, that's so not neat. Sorry. On the other side, you have, on the other strand, right, it's opposite, so it's going to be upside down. Right, here's your 5', 5', and then ends up with your 3' OH up top. I apologize for the lack of neatness on that. But, say 5' to 3', that direction, right, 3', or 5' to 3' that direction. They're anti-parallel. Parallel, but in opposite directions. They are a two-way street separated by a double yellow line. Okay? All right, the linkages between the two, and we'll talk more about this when we go through the structure and replication of nucleic acids. Nucleic acids? Man, this must be one of those days. All right, nitrogenous bases pair up with one another. Adenine pairs with thymine, cytosine pairs with guanine, adenine bonds with uracil when a DNA-RNA hybrid. All right, this is what we call Chargaff's rules, which leads to complementary pairing. Right, Chargaff was a scientist who looked at the composition of DNA and figured out that adenine, the percentage of adenine always equal the percentage of thymine, and cytosine always equal the percentage of guanine, and then came up with the complementarity bases from there. Okay, so complementary pairing: A to T, C to G. All right, thymine is not present and goes. All right, here's a structure of double helix. Your sugar phosphate backbone is just illustrated as a ribbon structure to show your bases. And then you have your transfer RNA, which also has secondary structure. Okay, has structure because of base pairing between some of the nucleotides, so G to C, A to U, and gives this secondary structure. Parts that are important for this part up here is not bound, doesn't have complementary, and then down here also does not have a complementary base pairing, doesn't have a double-stranded structure. Okay, this is still one single strand that is folded up on itself. There's not a second strand there. There are some aspects of double-stranded parts of it, but that's because it's binding between two parts of the same strand. Now, what does this mean for evolution? DNA and proteins as evolution. How do we compare? Why do we compare? How do we know? How do we use DNA and proteins as evidence to show that evolution is occurring or has occurred? Or how related one organism is to another. Okay? Chimpanzee DNA and human DNA is about 98% identical. Okay? Meaning the genes we have, the sequence of the DNA bases, A, T, C, G, we compare one gene, hemoglobin, in humans to hemoglobin in chimps, and they're pretty close. Okay? Why? We're a close relative. We are not necessarily the same family, but we are close relatives. You then compare to outside families. You compare 4 different mammals. Hemoglobin of 4 different mammals that are land breathing. And then you have sequence homology, and what we call sequence comparison. Sequence identity. They are alike. They are identical, meaning that they probably have similar function. Gives you idea about their function of a protein and the relatedness of them. All right, and this is all molecular. Okay, here's an example of hemoglobin. All right, between a human, a monkey, and a gibbon. You see any differences? Any others?
Yeah. The first one at the end.
Okay, just to illustrate 5 different differences that you see between amino acids. Does that change? What does that tell you? Are these organisms closely related?
Yes.
Probably. Okay. If I was to put up one from a dolphin up here, you might see 25% identical, and there are lots of differences. So that would tell you that the monkey, the gibbon, the human are more closely related to each other than they are to a dolphin. It gives you evidence for relatedness, how close, right? How close they are related, where they've shared an ancestor, and so on. All right, so review your concept. At the end of your chapter, you have this chart, these 2 charts, or this chart, and it has this first part, second part. Use this as a great study guide. Okay, it breaks down everything you need to know about these macromolecules into an easy reference. Examples, functions, and it even puts in the monomers for you. Okay, what they are. So you say examples, your monosaccharides, disaccharides, polysaccharides, right? The components, your monosaccharide monomer, right? And he breaks down glycerol, phospholipids, steroids. There's components of proteins and nucleotides, examples, and even the function of them. Great little study concept check at the end of the chapter. Questions on chapter 3. On Friday, we'll start chapter 4, and we'll probably get through quite a bit of it in one day. So be prepared to go pretty quick on Friday. And again, your test grades will be posted on Moodle this afternoon.